We are looking at the machinery that allows you to know that the out there even exists. We are talking about the architecture of touch.
It is a fascinating shift in perspective. We spend every single waking second of our lives interacting with the world, touching things, holding things, just moving through space. But we so rarely stop to consider the physiological orchestration that actually allows that interaction to happen.
I want to start with a little experiment for everyone listening. Wherever you are right now, unless you are driving a car and need both hands on the wheel, please, I want you to just pause for a second, pick up a coffee mug, or touch the fabric of your pants, or just run your hand over the armrest of your chair.
Okay, I'm doing it. I'm touching the desk right here.
It feels simple, right? It's just feeling. You touch the desk, you know it's smooth, you know it's hard. End of story. It feels immediate and totally uncomplicated. But according to this incredibly dense source text we have, chapter 48 on somatic sensations, that simple act is actually a massive illusion.
That is the perfect way to put it. It is an illusion of simplicity. What you perceive as touching a desk is actually millions of sensors firing, high-speed data highways transmitting these encoded signals, and a brain that is actively filtering, sharpening, and really reconstructing reality in real time.
Wow.
It is a masterpiece of biological engineering that makes our most advanced robotics look like, well, like children's toys.
So, our mission today is to unpack this masterpiece. We are going to strip away the skin, metaphorically, of course, and look at the wiring underneath. We're going to go line by line through the mechanics of how we feel. And I promise, by the end of this hour, you are going to look at your own fingertips with a whole new level of respect.
I'm ready. Where do we begin?
Well, we need to define the scope first. The text uses this term somatic sensations. I think for a lot of us, when we hear senses, we think of, the big five from kindergarten: vision, hearing, smell, taste.
And touch. But touch is doing a lot of heavy lifting in that list. In neurology, we make a clear distinction between the somatic senses and the special senses.
Okay.
So the special senses are things like vision, hearing, smell, taste, and equilibrium, your sense of balance. Those are highly localized. You hear with your ears, you don't hear with your elbow.
Right, makes sense.
But somatic comes from the Greek word soma, which means body. So these are the senses that come from everywhere, from the skin, the muscles, the joints, the deep tissues. It's the data feed from the entire physical frame.
And the text breaks that down even further, doesn't it? It's not just one big body sense.
No, not at all. It classifies somatic senses into three pretty distinct physiological types. First, and this is our main focus for this deep dive, you have the mechanoreceptive somatic senses.
Mechanoreceptive?
Yeah, that covers tactile sensations, so touch, pressure, vibration, and also position sensations, which is knowing where your limbs are in space.
Okay, so that's the architecture of touch and knowing where your body is. What are the other two?
The second is the thermoreceptive senses, which is just detecting heat and cold. And third is the pain sense, which is activated specifically by tissue damage.
It's interesting that pain is its own category. I have to admit, I always thought pain was just too much touch or too much heat.
That is a very, very common misconception. Pain isn't just an overload of the touch system. It is a completely separate alarm system with its own wiring, its own rules, its own highways. But for today, yeah, we are going to laser focus on that first group, the mechanoreceptors. We want to understand the texture of the world.
Perfect. So let's dive into what the outline calls Section 1, the cast of characters.
The receptors.
Before reading this, I kind of just thought, a nerve is a nerve. You touch something, the nerve gets squished, it fires, the brain says poke. Simple. But looking at this text, the skin is packed with these very specific, very different types of sensors. It's not a generic workforce, it's a specialized team of experts.
It really is. Think of it like a construction crew. You don't just have workers. You have electricians, plumbers, drywallers, supervisors. They all look at the same building, but they see completely different things.
That's a great analogy.
The skin is the same. It has 6 specific types of tactile receptors that the text outlines, and each one is tuned to a different aspect of physical reality.
Let's meet the team then. I want to start with the most basic one. The text calls them the free nerve endings.
These are exactly what they sound like. They are the generalists, the grunts, if you will. They are found everywhere in the skin and in many other tissues. A great example the text gives is the cornea of the eye.
The eye? You mean the surface of my eyeball?
Yes. The surface of your eye contains only free nerve endings. There are no specialized sensors there at all. Yet if you touch your eye, which please don't do that experiment, you can feel touch and pressure.
Oh, yeah. It's incredibly sensitive.
Exactly. So these simple, unspecialized endings can detect touch and pressure. But they are crude compared to the others. They can tell you something is happening, but they aren't great at telling you exactly what is happening or, the fine details of the texture.
So they are the infantry, found everywhere, doing the basic job of alerting you to contact. But then we get to the specialists. Let's talk about the Meissner's corpuscles.
Now we're getting into high fidelity.
Good.
Meissner's corpuscles are these fascinating little structures. If you look at the diagram in the text, these things look like, well, imagine a microscopic balloon sort of elongated and just stuffed full of these tangled branching wires.
And these aren't everywhere, right? The text makes a point about that.
No, and that's the critical distinction. They are found almost exclusively in what's called glabrous skin.
Glabrous. That's a great word.
It just means non-hairy skin. Think about your body for a second. Most of it is covered in fine hair, your arms, legs, your back. But your palms, the soles of your feet, your lips, those are naked.
Right.
That's where the Meissner's corpuscles live,
Hmm.
in those high-traffic, high-information areas.
Okay, so they're in the high sensitivity zones, my fingertips. What are they actually doing down there?
They're detecting texture and light touch. But the way they do it is really clever. The text says they adapt in a fraction of a second.
I read that part, but I wasn't entirely sure what adaptation meant in this context. It sounds like they just get used to the feeling and stop working.
That is actually exactly what they do, and it's a feature, not a bug.
Wait, how is stopping working a feature? If I'm touching something, don't I want to know I'm touching it?
You do, but you don't need to be yelled at about it constantly. Think about it this way. Put your hand flat on your desk and hold it perfectly still.
Okay, doing it. Hand is on the desk.
You felt the death when your hand landed, right? You felt that initial contact.
Sure, I felt the impact, the temperature.
But right now, 5 seconds later, do you really feel the desk? I mean, are you actively processing the sensation of deathness? Or has it kind of faded into the background?
It's definitely faded. I mean, I know it's there, but I'm not really feeling it anymore.
That is the Meissner's corpuscle shutting up. It fired when you made contact, told the brain, we've touched a surface, and then it adapted. It went silent to save bandwidth.
Okay, so it's lazy.
It's efficient, because watch what happens when you move your hand. Slide it across the desk.
Okay, yeah, now I feel everything. The grain of the wood, a little bit of dust, the temperature difference is more noticeable.
Exactly. Because you are moving, the skin is vibrating slightly as it hits those microscopic imperfections. The signal is changing. And Meissner's corpuscles love change. They fire like crazy when the stimulus changes, which is why they are perfect for telling you if something is smooth or rough or sticky or slippery. They are texture detectives.
That makes so much sense. That's why when we want to check the quality of fabric, we rub it between our fingers. We don't just pinch it and hold still. We rub. We are activating the Meissners.
Precisely. You're giving them the changing signal they crave.
Okay, next up in the lineup, Merkel's discs.
Merkel's discs are often found in the same areas as Meissner's, like the fingertips, but they have a totally different personality. They are often grouped together in something the text calls an igodome receptor, or a touch dome under the skin.
A touch dome. Sounds slightly sci-fi.
It's essentially a little upward projection of the skin epithelium. But here is the key difference in function. Merkel's discs transmit an initially strong signal, but then they adapt partially and settle into a weaker continuous signal. They don't turn off completely like the Meissner's.
So using the table analogy, Meissner tells me I touch the table, then goes silent. Merkel tells me I touch the table and then keeps whispering, "You are still touching the table. You are still touching the table."
Exactly. That's a perfect way to describe it. They provide what are called steady-state signals. This is absolutely critical for determining continuous touch against the skin.
Why do we need that if we have Meissner's to tell us about the initial contact?
Imagine you are holding a pen or your phone. You aren't constantly rubbing the object, you're just gripping it. If you only had Meissner's corpuscles, the moment your hand stopped moving, you would effectively lose the sensation of the pen. You might drop it because your brain would think it disappeared. Merkel's discs keep firing that low-level continuous signal to tell your brain the object is still in the grasp. We're still holding on.
Got it. So we have the change detector, which is Meissner, and the steady state detector, which is Merkel. What about number 4? The hair end organ.
This is one we often overlook because we are so focused on our fingertips.
Yeah.
But we have hair over most of our bodies. The hair end organ is a nerve fiber that wraps around the base of the hair follicle.
So the hair itself isn't the nerve. It's not alive?
No, the hair is dead protein. But it acts as a lever. It's a mechanical amplifier. The slightest movement of any hair on your body stimulates that nerve fiber wrapped around its base.
Right, I've noticed this. If a mosquito lands on my arm, barely touching the skin, I feel it instantly because it must have brushed against a hair.
That is the hair end organ in action. It detects movement of objects on the surface of the body or just the initial contact. It is basically a highly sensitive proximity alarm. And like Meissner's corpuscles, it adapts very readily. It detects the event of contact. It's saying, incoming, something is here.
It's the burglar alarm of the skin. Okay, moving deeper into the tissue now. Number 5, Ruffini's endings.
Now we're going into the deeper layers of the skin and even into internal tissues like joint capsules. Ruffini's endings are multi-branched and encapsulated, but their superpower is that they adapt very, very slowly.
Even slower than Merkel.
Much slower. They are designed to signal continuous states of deformation. Think about heavy, prolonged touch or pressure. If someone grabs your arm firmly and just holds it, that deep, sustained sensation is largely due to Ruffini's endings.
And you mentioned they're in joint capsules. That seems important.
Yes, this is crucial for proprioception, which is just the fancy word for knowing where your limbs are.
Yeah.
Because they don't adapt, they can tell you about the continuous rotation of a joint. If you have your arm bent at a 90-degree angle for 5 minutes, your brain knows it is still bent. That is largely thanks to receptors like Ruffini's endings that just keep firing, just keep saying, still bent, still bent, still bent.
They are the endurance runners of the receptor world. Okay, and finally, number 6. The text describes these as the specialists for rapid changes, the Pacinian corpuscles.
My personal favorites.
And my
These lie deep in the skin and the fascial tissues. If you look at them under a microscope, they look like little onions. They have all these concentric layers.
And an onion.
Yeah. And they are the specialists for speed. They adapt in hundredths of a second.
Hundredths of a second. That is incredibly fast.
It's almost instantaneous.
Yeah.
They are stimulated only by rapid local compression. So a steady pressure does nothing to them. Because they reset so quickly, they are critical for detecting one thing in particular, tissue vibration.
Vibration, so like if I'm holding a power drill or something.
Exactly. If you are using a power tool where you feel the hum of an engine through the steering wheel, or even the feeling of a low rumble from an earthquake, that is the Pacinian corpuscle vibrating in resonance with the stimulus. It can fire hundreds and hundreds of times per second.
So just to recap the team here, because it's a lot to keep track of, we have Meissner for texture and light touch on fingertips, Merkel for continuous touch holding the pen, hair end organs for surface movement, the mosquito alarm, Ruffini for deep pressure and joint position, and Pacinian for vibration.
And the free nerve endings, the generalists, for everything else in between.
It's an amazing toolkit. But here's the thing, having the sensors is only step 1. It doesn't matter if the Pacinian corpuscle detects a vibration if that signal doesn't get to the brain. We need wires. And this brings us to section 2, the wiring system.
Right. And here is where we see that the body is very, very economical. It's actually quite stingy with its resources. It doesn't use the expensive wiring for everything.
The text distinguishes between different types of nerve fibers based on their speed and size. We have these type A beta fibers and then the smaller A delta and C fibers. Can you break down the difference?
Think of type A beta fibers as your high-speed fiber optic cables. They are large, they are myelinated, which means they're wrapped in this fatty insulation for speed, and they transmit signals at velocities ranging from 30 to 70 meters per second.
That is just absurdly fast. I'm doing the math in my head. That's over 150 miles per hour in some cases.
It is, and almost all the specialized receptors we just discussed, Meissner's, Merkel, Pacinian, Ruffini, they use these A beta fibers.
Why? Why do they get the fast lane?
Because the information they carry requires precise localization and rapid analysis. If you were trying to identify a texture with your finger, or catch a ball, or adjust your grip on a tool, your brain needs that data now. Milliseconds matter for motor control and for detailed perception.
So that's the premium service. Contrast that with the free nerve endings.
Right. The free nerve endings, the generalist, they generally transmit via small type A delta fibers, which are slower, maybe 5 to 30 meters per second, or the unmyelinated type C fibers, which just crawl along at 0.5 to 2 meters per second.
2 meters per second? That's walking pace. That is incredibly slow for a nerve impulse.
It is, but these fibers are carrying cruder signals. Things like dull pressure, poorly localized touch, and especially, as the text points out, tickle.
Tickle. Tickle gets the slow bus.
Yes. Think about it. Do you need to know the exact microscopic location of a tickle the instant it happens? Or do you just need a general nagging sense that something is crawling on you?
I guess a general sense is fine. But why not just make everything fast? I mean, if evolution is about survival of the fittest, wouldn't the fastest nervous system win? Why keep the slow wires at all?
It's a space issue. It's a bandwidth management issue. Fast fibers are fat. They take up a lot of room in the nerve bundle because of that thick myelin insulation. Slower fibers are tiny.
Like the difference between a huge industrial power cable and a tiny little headphone wire.
That's a great way to think about it. If all your sensory nerves were A beta fibers, those giant cables, your spinal nerves would be enormous. They literally wouldn't fit through the holes in your vertebrae. You'd have a spinal cord as thick as your arm.
So the body prioritizes. It makes a trade-off.
It has to. Critical, spatial, rapid data gets the high-speed, high-cost fiber optic line. Cruder, less urgent data gets the slow lane to save space.
That is fascinating. Speaking of tickles and vibrations, let's move to Section 3, Special Senses. The text dives a bit deeper into vibration, tickle, and itch specifically.
Vibration is an interesting one because, as we touched on, vibration isn't really a separate sense. It is just repetitive touch signals being fired very quickly.
Right. And the text says different receptors handle different frequencies, which makes sense.
It goes back to their adaptation rates. Pacinian corpuscles, the onion-like ones, can handle extremely high frequencies. The text says 30 to 800 cycles per second. Because they reset so fast, they are ready to fire again almost instantly.
So that's the high-pitched buzz of a dentist's drill or something.
Yes, exactly. Whereas Meissner's corpuscles adapt a bit slower, so they handle the lower frequencies. The text puts it at 2 to 80 cycles per second. Think more of a low rumble.
So together, they covered the whole spectrum of buzzing and humming.
Correct. Now, tickle and itch are different beasts entirely. The text is very specific here. These sensations are detected by very sensitive, rapidly adapting, mechanoreceptive-free nerve endings. And this is key. They're located almost exclusively in the superficial layers of the skin.
That explains why you can relieve an itch by scratching just the very surface. You don't have to dig deep to get it.
Right, and they transmit via those slow, pokey type C fibers we mentioned. The evolutionary purpose here is really compelling. Why do we itch?
To annoy us.
It certainly feels that way sometimes. But the text suggests the purpose is to call attention to very mild surface stimuli like a flea or a mosquito or a crawling ant in order to activate the scratch reflex.
So it's a defense mechanism against parasites and insects?
It is. And here's the cool part about the scratch reflex. Have you ever wondered why scratching actually stops the itch?
I always just assumed you were physically scraping the bug off or something.
Well, that's the functional goal, sure.
Right.
But physiologically, scratching relieves the sensation of itch because scratching creates a mild pain signal. And pain signals actively suppress itch signals in the spinal cord. It's a process called lateral inhibition.
Wait, so pain cures itch. You're fighting fire with fire.
In the wiring of the spinal cord, yes, they are antagonistic signals. The pain signal effectively jams the frequency of the itch signal. It shouts it down.
That explains why if you have a really bad mosquito bite, sometimes you scratch it until it almost hurts or you slap it and it actually feels better. You're trading the maddening itch for a clean, simple pain signal.
Exactly. The brain prefers the pain signal over the itch signal in that context. It silences the itch channel for a moment.
That is wild. Okay, so we've got the receptors, we've got the wires. Now we need to look at the roadmap. Section 4, the two great highways.
Now, this is where the engineering gets really impressive. We have these millions of signals coming from the Meissners, the Pacinians, the hair follicles. They all have to get to the brain.
It sounds like a traffic jam waiting to happen.
It would be if the body didn't have a very, very strict traffic control system. The text describes two main highways going up the spinal cord to the brain. And they're completely different systems.
Different how? Like different speeds?
Different speeds, different structures, and they carry completely different types of cargo. I like to think of them as the high-speed, perfectly paved interstate and the winding, bumpy back alley.
Okay, walk me through the interstate first.
The technical name is a mouthful. The dorsal column medial lemniscal system. Let's just call it the DCML.
DCML, the interstate. Got it.
Right. This system is built for speed and precision. It uses those fat, myelinated A beta fibers we talked about earlier.
The Ferrari lane?
Exactly. Transmitting at up to 110 meters per second in some cases. But more importantly, it is incredibly organized. It has a high degree of spatial orientation. It preserves the map of your body perfectly.
What does that mean, preserves the map?
It means if a nerve fiber enters the spinal cord from your thumb, it travels up the cord in a very specific dedicated spot.
Right.
The fiber from your index finger is right next to it. The middle finger next to that. They don't mix. They stay in their lanes all the way up to the brainstem.
So the spatial relationship is physically maintained. The thumb wire never gets crossed with the toe wire.
Precisely. That's why you can close your eyes and know exactly down to the millimeter where a mosquito just landed on your arm. That spatial data is expensive to maintain, but the DCML system does it flawlessly.
Okay, so that's the premium package. High speed, perfect tracking. What kind of information gets this VIP treatment?
The text lists them out. Touch requiring fine localization, fine gradations of intensity, vibration, movement against the skin, and position sense from the joints. Basically all the high-res data.
And there's a twist in the road, isn't there? The text mentions decussation.
Yes, the decussation, the crossing. The DCNL signals shoot straight up the spinal cord on the same side they entered. So a left-hand signal goes up the left side of the spine, but when they hit the medulla, which is in the lower brainstem, they synapse and then cross over to the opposite side.
So the left hand eventually talks to the right side of the brain.
Correct, but not until it gets all the way up to the brainstem.
Okay, so that's the interstate. What's the other option? What's going on in the back alley?
The anterolateral system.
That doesn't sound as promising.
Well, it's much older, evolutionarily speaking. It uses thinner wires, many of them unmyelinated. It's slower. The text says up to about 40 meters per second, so less than half the speed of the other system. And the map is, well, the spatial orientation is poor. It's a bit fuzzy.
So why do we keep it? Why not just upgrade everything to the DCML interstate?
Because the back alley handles the things the interstate won't touch.
Such as?
Pain, thermal sensations, so extreme heat and extreme cold, crude touch and pressure, itch, tickle, and even sexual sensations.
Oh, the primal stuff, the really important survival and emotional signals.
Exactly. The things that don't necessarily need millimeter precision, but they need to get there eventually, and they trigger deep, powerful responses. And here is the crucial anatomical difference. These fibers enter the spinal cord and cross to the opposite side immediately.
Oh, interesting. So DCML stays on the same side until the brainstem, but the anterolateral system crosses right away in the spine.
Correct. It crosses through a structure called the anterior commissure and then ascends up the anterior and lateral white columns of the cord. This creates a very specific clinical picture if the spinal cord is damaged.
How so?
Think about it. If you have an injury that cuts through just the left half of your spinal cord in your lower back, what happens to the sensations from your legs?
Okay, let me think this through. The DCML, the fine touch, from the left leg is traveling up the left side. So that signal gets cut off by the injury. So I lose fine touch on my left leg.
Right. What about pain?
But the pain signals from the left leg, they entered the cord and crossed over immediately to the right side. So they would bypass the injury on the left side and keep going up.
Exactly.
Yeah.
But what about the pain signals from the right leg?
The pain signals from the right leg crossed over to the left side, right where the cut is, so they get blocked.
So what's the result?
So I lose fine touch on my left leg, but I lose the sense of pain and temperature on my right leg
Right.
from the same injury.
Bingo. This dissociation of sensation, it's called Brown-Séquard syndrome, is a classic diagnostic pearl for neurologists. It tells them exactly where a lesion is located based on which senses are missing from which side of the body.
That is fascinating. The separation of these pathways isn't just trivia, it's how doctors can map injuries inside the spinal cord without even looking. But eventually, both roads lead to the same city, the somatosensory cortex. Which brings us to Section 5.
The destination. The text describes the somatosensory cortex as being located in the postcentral gyrus. It sits right behind that big central fissure of the brain. And this is where it introduces the concept of the homunculus, the little man, the body map.
The homunculus is famous. Even if you haven't studied anatomy, you've probably seen that weird drawing. It looks like a distorted, almost grotesque little man draped over the surface of the brain.
Yes, it is a visual representation of how much brain power, how much cortical real estate is dedicated to processing sensory information from different body parts. And as you said, it is severely distorted.
Right. In the pictures, the hands and the lips are absolutely enormous. And the entire trunk and the legs are these tiny little things. Why is that?
The text explains that the size of the area in the brain is directly proportional to the number of specialized sensory receptors in that body part, not the physical size of the body part.
So because my lips and my fingertips are packed with Meissner's corpuscles and Merkel's discs and all those other high-res sensors, they get a huge chunk of real estate in the cortex.
Correct. A huge amount of processing power is needed to interpret all of those millions of signals. Meanwhile, The skin on your back has relatively few specialized receptors, so it gets a tiny little sliver of the cortex.
It really shows what the brain prioritizes. It cares about manipulating the world with our hands and eating and speaking with our mouth. The back is just there to hold everything together.
Pretty much. Now let's zoom in on the cortex itself. The text talks about layers and columns. It says there are 6 layers of cells.
I found this part a bit dense. Can you simplify the layers for us?
Sure, think of the cortex as a 6-story building. The main sensory highway, the DCML system, pulls up to the building, and the signals get off the elevator on the 4th floor. So layer 4 is the primary input layer.
Okay, so the signal arrives in the middle.
Right. From there, the information spreads up and down to the other floors for processing. Layers I and I, the top floors, receive more diffuse, nonspecific input from lower brain centers that kind of sets the general tone. And layers V and VI, the basement floors, are the output layers. They send axons back down to deeper parts of the nervous system to control the signal flow.
So input comes in the middle, spreads up for detailed processing, and goes down for feedback and control.
That's the basic idea. But the more interesting structural point to me was the concept of the vertical columns.
Yeah, what is that about?
This is absolutely key to understanding how the brain organizes data. The neurons aren't just a random mush. They are arranged in these tightly packed vertical columns, about 0.3 to 0.5 millimeters in diameter. Each column contains maybe 10,000 neurons. And crucially, each column is dedicated to a specific sensory modality.
A modality. So you're saying one column is for joint stretch, and the one right next to it is for hair touch.
Exactly. It's like a filing system. You don't just have a finger area in your brain. Within that finger area, you have a specific column of neurons that only cares if the finger joint is bending. Right next to it, there's a column that only cares if the skin on that finger is vibrating. And next to that, a column that only cares about deep pressure.
It's incredibly organized. It's not a soup of neurons. It's a precise computational grid.
It is a highly structured computational machine.
But what happens when this machine breaks? Section 6 covers the functions and failures of somatosensory area I. What happens if we were to, say, cut it out?
An excision, yes. If you were to surgically remove somatosensory area I, the result is actually quite surprising. You don't lose all sensation.
You don't. I would have assumed you'd go completely numb in that part of the body.
No. The text says you can still feel crude pain and temperature. You can still feel that something is touching you.
So the rudimentary stuff, the basic feelings, must happen lower down in the brain.
Yes, in the thalamus and the brainstem. Those ancient, more primitive parts of the brain can handle the basics like ouch or hot. But without the cortex, you lose all the details. You lose distinct localization. You can't tell exactly where you were touched. You lose the ability to judge critical degrees of pressure or the weight of objects.
And the text mentions a specific condition, asterognosis.
Asterognosis.
Right.
It's the inability to judge shapes or forms using touch. Imagine this. I put a key in your hand and ask you to close your eyes. If your cortex is intact, you roll it around, you feel the texture, the temperature, the shape, and you say, it's a key.
Because I can feel the teeth, the cold metal, the shape of the head, and I put it all together.
Right. But if you have asterognosis, because you don't have somatosensory area, you would feel metal object, maybe hard, maybe cold, but you could not synthesize those individual feelings into the complex concept of key. You would be holding it, but your brain wouldn't know what it is.
So that distinction between feeling and interpreting is crucial. The cortex is what turns raw data into a recognizable object.
Precisely. It turns sensation into perception.
And it gets even weirder in Section 7, the somatosensory association areas.
Yes, Brodmann's areas 5 and 7. These areas sit right behind the main sensory cortex. Their job is to decipher meaning. They take the processed info from the somatosensory cortex, and they combine it with data from the thalamus, the visual cortex, and the auditory cortex to create a complete, complex picture of your body in space.
And the failure here is called amorphous synthesis.
This is one of the most bizarre and fascinating conditions in all of neurology. If you remove the association area on one side, say, the right side of your brain, the person loses the ability to recognize complex objects or forms on the opposite, the left side of their body. But it goes further than that. They essentially forget that the opposite side of their body even exists.
This is the hemineglect phenomenon, isn't it? I've seen videos of this.
Yes. A patient might only dress the right side of their body. If you ask them to draw a clock, they might crowd all 12 numbers onto the right side of the circle and leave the entire left side completely blank. They lose the concept of the left side of space.
That is just mind-blowing. It's not that they can't see it or feel it, it's that their brain has stopped processing it as part of their reality.
It shows that feeling isn't just a passive input, it's an active mental construct. We build the world in our heads. And if the construction crew, areas 5 and 7, goes on strike, that part of the world ceases to exist for us.
You literally lose half of your reality. That is terrifying and amazing at the same time.
Indeed.
Let's shift to Section 8, signal processing. How does the brain take all this incoming noise and make it sharp and clear? We have to talk about the two-point discrimination test.
This is a classic neurological test. You take two sharp points, like two needles or the ends of a paperclip, and press them lightly against the skin. Then you ask the person, are you feeling 1 point or 2?
The text gives some pretty starkly different numbers. On the fingertips, it says you can distinguish points that are just 1 to 2 millimeters apart.
Right. Your fingers are incredibly high resolution. But what about on your back?
The text says 30 to 70 millimeters. That's a huge difference.
It is. That's nearly 3 inches. The points have to be 3 inches apart on your back before you realize it's two distinct points. Otherwise, it just feels like one big blurry poke.
Why the huge difference? What's going on there?
Well, part of it is simply the number of receptors, the
Yeah.
pixel density, if you will. Your fingertips are packed with them. But the brain also uses a very clever trick called lateral inhibition to sharpen the image.
Lateral inhibition. This sounds like a strategic military maneuver. How does it work?
This is one of my favorite concepts in the whole text because it shows how the brain essentially lies to us in order to help us see the truth more clearly.
That sounds like a paradox.
Think about it like this. Imagine you poke your skin with a single needle point. In reality, your skin is flexible, right? It's soft tissue. It bends.
Sure, it's not a hard plastic shell. It dimples when you poke it.
Right. So obviously the receptor directly under the needle point gets stimulated the most. It's firing at 100%. But the skin around that point is also being stretched and pulled by that dimple. So the receptors next door, they're also getting stimulated, maybe at 50%. And the ones a little further out at 20%.
So the signal going to the brain shouldn't be a sharp point. It should look more like a fuzzy hill with a peak in the middle and sloping sides.
Exactly. If the brain just listened to the raw, unprocessed input, every single poke would feel like a blurry blob. You wouldn't be able to read Braille or feel the sharp edge of a piece of paper or locate a tiny splinter.
So how does lateral inhibition fix that blob?
It creates a winner-take-all scenario. The central neuron, the one that's screaming at 100% intensity, doesn't just send a signal up to the brain. It also sends little side branches to its immediate neighbors.
To the 50% guys.
Yes. And these side branches are inhibitory. They release a chemical that essentially tells the neighboring neurons to shut up.
Wow, so the loudest guy in the room is actively gagging the people standing next to him.
That's a slightly aggressive way to put it, but yes, that's the mechanism. It effectively silences the background noise. It shaves the size off that fuzzy hill of sensation until only the sharp peak remains to be sent to the brain.
So the sensation of a sharp point is actually a neurological illusion. The brain creates the sharpness.
It is a computed contrast. The brain artificially enhances the sharpness by actively deleting the fuzziness around the main signal. It's exactly like the sharpen filter in a photo editing program.
So the brain is actively manipulating the data to make it clearer than it actually is. The text also brings up some math here regarding how we perceive intensity. The Weber-Fescher principle versus the power law.
Right, this is about how we judge the strength of a signal. Weber and Fechner proposed that our detection of intensity is logarithmic.
Meaning what in simple terms?
It means it's all about ratios, not absolute values. The example the text gives is great. If you were holding 30 grams, you could probably notice if I add 1 more gram. But if you were holding 300 grams, adding that same 1 gram will feel like nothing. You need to add 10 grams to notice a difference. The ratio stays constant.
Okay, that makes sense intuitively. We notice relative change, not absolute change.
Exactly.
Yeah.
But the text says that the power law is actually a more accurate description for a wider range of stimuli. The formula looks complicated. It's something like interpreted signal strength equals K times stimulus minus K to the power of Y.
I won't ask you to solve for why.
The math itself doesn't matter as much as the concept. And the concept is that there is a predictable mathematical relationship between the interpreted stimulus and the actual stimulus. Our brain compresses reality mathematically so we can handle an enormous range of sensations, everything from a feather's touch to a heavy blow, without overloading the circuits.
We are running biological compression algorithms on reality. That's amazing. Let's jump to Section 9. Position senses. Proprioception. How do I know where my arm is right now without looking at it?
There are two subtypes here. Static position sense, which is knowing the orientation of your body parts while you're still, and rate of movement sense, also called kinesthesia, which is feeling the speed of that movement.
And the receptors here are different from the skin ones.
It's a mix, but some key players come to the forefront. Muscle spindles are the VIPs here. The text says they're the most important receptor for determining joint angulation, especially in the mid-ranges of motion. They are constantly telling the brain how stretched or contracted each muscle is.
And what about at the extremes, like if I bend my arm all the way back until the joint locks?
Then other players kick in. The Pacinian corpuscles and the Ruffini endings in the joint capsules, along with Golgi tendon organs, become very active. They're specialized to detect the extremes of angulation and also the rapid rates of change.
There is a really cool detail in the text about how the thalamus processes this information. It says individual neurons in the thalamus are actually tuned to specific angles of a joint.
Yes, this is just incredible. Researchers could put an electrode into the thalamus, and they found neurons that would fire maximally when an animal's joint was at full rotation, and other neurons right next to them that would fire maximally when it was at minimal rotation.
So the brain isn't just getting one signal for bend.
No, it's getting a whole pattern of signals.
Yeah.
The brain basically looks at the graph. It compares which populations of neurons are shouting and which ones are whispering to triangulate the exact angle of the joint down to a fraction of a degree.
It's basically digital encoding. Neuron A is firing at 80%, neuron B is at 20%, therefore the arm must be at a 45-degree angle.
Precisely. It's pure computation.
We glossed over the anterolateral pathway earlier, the back alley. But Section 10 does a deep dive, and I want to circle back to where these particular signals end up.
All right, so remember, the AL pathway is carrying pain, temperature, crude touch. It enters the dorsal horn, the spinal cord, crosses immediately, and heads up. But unlike the fine touch system, which has a direct express route to the sensory cortex, the AL system makes some different stops.
The text says only a small fraction of its fibers go to that same specific part of the thalamus, the ventrobasal complex, where the main touch signals go.
Correct. Most pain signals, for instance, terminate in two key areas: the reticular nuclei of the brainstem and the intralaminar nuclei of the thalamus.
Why does that matter? What do those areas do differently?
The reticular formation is essentially the brain's alarm clock. It controls wakefulness and alertness. This is why a strong pain signal jolts you awake. It's why you can't sleep when you have a toothache. The signal is hardwired into the arousal and alarm center of your brain, not just the location mapping center.
Ah, so the DCML system tells you you are being touched with a pin on your left index finger. The AL system is screaming, something is wrong. Wake up. Pay attention.
That's a great way to put it. These areas handle the suffering aspect of pain, the emotional, affective, and arousal components, separate from just the raw sensation.
That is a profound distinction, the sensation of pain versus the suffering of pain.
It is. You can theoretically have the pure sensation of pain without the suffering if these pathways are disconnected, which tragically happens in some old types of lobotomies, but normally they are inextricably linked.
Okay, moving to Section 11, corticofugal signals. Corticofugal sounds like a magic spell.
It does. It just means fleeing the cortex. These are signals that are traveling backward from the brain down to the lower sensory stations.
So the brain talks back to the hand. I thought it was a one-way street.
We usually think of sensory systems as one-way streets, from the hand to the brain. But the text makes it clear that signals are constantly coming down from the cortex back to the thalamus, the medulla, and even the spinal cord. And they're almost entirely inhibitory.
Inhibitory. So the brain is telling the sensors to be quiet. Why would it want to inhibit the incoming signal?
Two main reasons. First, to decrease lateral spread, that sharpening effect we talked about with lateral inhibition. The brain can send signals down to fine-tune that process, to tell the peripheral neurons to focus even more. And second, to keep the sensory system operating within its proper range.
Like an automatic gain control on a microphone.
That's the perfect analogy. If the input is too loud, too intense, the brain sends an inhibitory signal down to dampen the sensitivity of the relay neurons so the system doesn't get completely overwhelmed and saturated. It actively manages its own volume knob.
So we are constantly tuning our own reception of the world based on what the brain thinks is important. That is incredibly sophisticated. Finally, Section 12, dermatomes.
The map of the spine on the skin. A dermatome is the segmental field of skin that is supplied by the sensory nerves from a single specific spinal nerve root. If you look at the dermatome map, the body is striped like a tiger, with each stripe corresponding to a spinal level.
C6, L5, S1. These are the spinal levels that doctors are always talking about.
Yes. But the key thing the text notes here is that there is significant overlap between adjacent dermatomes.
Overlap, so the stripes aren't clean lines.
No, the boundaries are very blurry. The L5 dermatome overlaps significantly with L4 above it and S1 below it. This is a crucial safety feature. It means if you sever one single spinal nerve root, you don't necessarily lose all feeling in that strip of skin because the neighbors on either side are able to pick up some of the slack. You'd have to lose multiple segments in a row to get total anesthesia in a region.
Redundancy. It's just good engineering.
It's vital engineering.
So let's try to bring this all home. We have gone from a single Meissner's corpuscle detecting a tiny ridge on a fingerprint, sending that signal up the high-speed A-beta highway, staying on the same side of the spine, crossing in the medulla, shooting up to the thalamus, arriving in layer CL of the somatosensory cortex, being sorted into a specific vertical column for texture, getting sharpened by lateral inhibition, and finally being interpreted as part of a key by the association areas.
It is a journey that happens in milliseconds, millions and millions of times a day, completely beneath our conscious awareness.
It really changes how I feel about, well, the simple act of feeling anything.
It should. And if I can leave you with one final thought from this entire deep dive, it connects back to that power law we talked about in the corticofugal signals.
Let's hear it.
We tend to think that our senses are like a high-definition video camera, just passively recording the world exactly as it is. But this text proves that is fundamentally false. Our senses are logarithmic. They are compressed. They are laterally inhibited to create artificial contrast. They are actively dampened and filtered by the brain's own control signals before we are ever consciously aware of them. What you feel is not the raw world. It is a heavily edited, photoshopped, and mathematically compressed reconstruction of the world that your brain has built for you. We don't just perceive reality. We biologically construct our own sensory reality from the ground up.
We construct our reality. That is a lot to process.
It keeps me up at night sometimes.
Well, on that note, I want to challenge everyone listening. Try the 2-point discrimination test on a friend or a family member. Take 2 pencils and poke them on the back and then on the fingertip. See the machinery in action for yourself.
And just take a moment to appreciate the millions of little sensors and the miles of wiring working perfectly so you can even do it.
Thanks for diving deep with us. We'll catch you on the next one.
Stay curious.